Dear learners, in this session we will deal with next part of this antimalarial agents. In the previous two sessions, we studied this definition and classification of antimalarial agents along with the life cycle of malaria and in detail the two classes, that is cinchona alkaloids and 4-aminoquinolines. Now in this session we will discuss the remaining classes of antimalarial agents, that includes 8-aminoquinolines, 9-aminoacridines, bigonides, sulfones, and this peroxides, isn't it? So there is a structural similarity between this quinine and 8-aminoquinolines, and also there is a structural similarity between this quinine and this quinoline 4-methanols. As you seen in this representation, the quinine contains this quinoline as a basic nucleus, and the part of this it form this parmaquine and pramaquine, that is 8-aminoquinolines. This is the 8 number of this 8-aminoquinoline, which is substituted with the amino alkyl chain will be developed, and these are the quinoline moiety is substituted with 8-aminoquinolines. These antimalarial drugs are based on the cinchona alkaloids, and this miscellaneous agents like mefloquines or this quinolone methanols, are the structures are also based on the cinchona alkaloid structure, that is quinine. These are the examples of 8-aminoquinoline, that is pramaquine, parmaquine, then pentaquine and isopentaquine. The structural similarity is that the methoxy group is substituted with this 6th position of this quinoline ring system. This, when we consider this 1, this is 2, this is 3, this one is 4, this is 5, this one is 6, this one is 7, and this one is the 8 number. The 8 number is substituted with this 6-methoxy, 8 number is substituted with the pentane. This is the pentane chain, this one is the pentane, and this 1st and 4th number of this carbon is substituted with the diamine functional group. So this is the IUPAC of this pramaquine, that is 4N 6-methoxyquinoline 8-eel pentane 1,4-diamine. It is that this is pentane 1, 2, 3, 4, 5. This 1 and this 4 number is substituted with this amine functional group. So this is known as 1,4-diamine. When we consider this pentaquine, this is again 6-methoxy, then this nitrogen, this is also nitrogen or amine functional group at 1, 2, 3, 4, 5, isn't it? So this also first, and this is first, and this is 5th number carbon is substituted with the amine functional group. So it is also a diamine, but this amine is substituted with isopropyl, isn't it? One hydrogen is replaced with the isopropyl functional group. So this is called as the pentaquine. This parmaquine also 1, 2, 3, 4, 5. It is also pentane, but it is diethylamine, isn't it? So it is what? Diethylamine. This is 6 again, methoxy, and 8 number is substituted with this what? 1. This is what? 1, and this one is the 4th number, diamine, but this 1 amine is substituted with the diethyl functional group. So this is parmaquine. When we consider this isopentaquine, this is also isopropyl group is substituted on this nitrogen or amine functional group. This is tafenoquine. It is the recent class drug of this 8-aminoquinoline, that is tafenoquine. When we consider this 1, the second number is substituted with this methoxy, isn't it? Then 3rd and 4th number is substituted with methyl, isn't it? 4. Then 5 number is with phenoxy, but that is what, isn't it? Trifluoromethane. And 6 number is also methoxy. This is also what? Amino, diamino, pentane, isn't it? 1, 2, 3, 4, 5, isn't it? So this is the dafenaquine under the heading of 8 amino quinolines. The mode of action or mechanism of action of this 8 aminoquinolines, it can generate reactive oxygen species via an autooxidation of 8 amino group, and the formation of this radical anion at 8 amino group has been proposed. As a result of this, it forms the cell destructive oxidants such as hydrogen peroxide, superoxide, and hydroxyl radical can form, leading to the oxidative damage to critical cellular components. So this is in short the mechanism of action of this 8 amino quinolines. The structural activity relationship of this 8 amino quinolines, when we consider the structures of this, so quinine or quinoline ring system is the important part, that is benzpyridine is important for biological activity. When we consider the 6 number is substituted with methoxy, it is important for biological activity. The first number is never been be substituted with any other alkyl or aromatic ring. So it when we substitute, it decreases the biological activity. When we increase the chain length of this pentane with the decrease or increase like 4 or 6 carbon atoms, it decreases the biological activity. When we replace this, or this nitrogen is a part of any aromatic or heterocyclic ring system, it, it is also not possible to form this antimalarial activity, or it generally not develop this antimalarial activity. When the groups are substituted on this second, third, fourth, it shows a pharmacological activity, but also the side effects are there. Like this dafenoquine is developed by substituting this methoxy group at the second position, but this third and the 7 number is not always been substituted with any other functional group or ring systems, isn't it? These are the SAR points of this what 8 amino quinolines. The next part is 9 amino acridines. So this 9 amino acridine examples are quinacrine and acriquine. This is more effective and less toxic than quinine. Also process the anticonvulsion activity. These uh, these drugs act at many sites within the cell, including interaction of DNA strands, the succinic dehydrogenase and mitochondrial electron transport and cholinesterase. So this is also this is what quinacrine. This is the acridine ring system. This is substituted with methoxy, and the amino group is substituted on this along with the chain of alkyl group. With the alkyl group is substituted on the amino function group at the chain, which is substituted on the amine, and the chloro group is substituted on this. So this is quinacrine. In case of this quinacrine, this D isomer is less toxic than L isomer. This is the quinacrine, then acriquine, and amino acriquine. These are the three structures of 9 amino acridine. This amino group is substituted on the 7th position of this. This is the 7th position. When the numbering system is starting from this 1, that is second number is on the methoxy. This is 1, this one is 2, this is 3, isn't it? This one is the 4, this is 5, this is 6, this one is 7, this is 8, this one is 9. So 9 amino acridine, and this one is the 10 number. So that's why this is called as a 9 amino acridine, means 9th position is substituted with the amine or amino functional group. This is called as the quinacrine or 9-amino acridine. The example quinacrine. It is it acts by many sites within the cell, including this interaction of DNA strands and the succinic dehydrogenase and mitochondrial electron transfer and cholinesterase. This quinacrine can cause the yellow discolorization of the skin and the urine, isn't it? Next one is pyrimidines and sulfones. The pyrimidines include the two drugs, that is pyrimethamine and trimethoprim, and sulfones, that is dapsone and sulfonamide. This dapsone is also used as an antileprotic agent, and sulfonamides, this chapter is also covered in last two lectures. Already covered the mode of action, SCRs, and the structure IUPAC along with this. This pyrimidines, this pyrimethamine and trimethoprim. Generally, the sulfonamides and trimethoprim are used as antibacterial. It is the combination, that is cotrimoxazole. The Ceptran DS is a trade name. When pyrimethamine is considered, this pyrimethamine and trimethoprim both and dapsone, these drugs having the same type of mode of action like sulfonamides. When it inhibits the dihydrofolate reductase enzyme, which is responsible for conversion of this dihydrofolic acid to tetrahydrofolic acid. When the sulfonamides, these drugs inhibit this dihydropteroate synthase enzyme, which is responsible for formation of this dihydrofolic acid. It will be generally reacting with para-aminobenzoic acid with pyridine, and it formed this dihydropteroic acid in presence of dihydropteroate synthesis. This is inhibited by the sulfonamide, and further this dihydropteroic acid is converted into dihydrofolic acid in presence of dihydrofolate synthase enzyme. This dihydrofolic acid is converted into this tetrahydrofolic acid, and this dihydrofolate reductase is inhibited by this pyrimethamine and trimethoprim type of drugs. Combinations are also used in this case, that is dapsone with trimethoprim, pyrimethamine, like sulfonamides with pyrimethamines are used as antimalarial agent. The next class is this is peroxide, artemisinin and artemether. These drugs are under the heading of peroxides. When we consider these naturally occurring artemisinin is the obtained from dry leaves of Artemisia annua, that is a sweet wormwood. This plant has to grow each year from seed because mature plants may lack the active drug, isn't it? The artemisinin series are the newest of the antimalarial drug and structurally unique when compared with the compounds previously and currently used. This is not structural similarity with the previously available drug like sulfonamides and this 4-aminoquinolines and 8-aminoquinolines like this. This all these structures are active against the Plasmodium genera that causes malaria, and the key structure appears to be trioxane. Isn't it? This contain a trioxane consisting of endoperoxide and dioxazepine oxygens. This is shown by somewhat this 3-aryl trioxanes at this trioxanes is there. Although this victims or erythrocyte, the malarial parasite consumes this hemoglobin consisting of this ferrous. The mode of action of this artemisinin and this artemether, it consumes in the victim's erythrocyte. The malarial parasite consumes the hemoglobin consisting of ferrous ion, converting into the toxic hematine containing ferric, and then reduced to heme with its ferrous ion, and the heme ion reacts with the trioxane moiety, releasing the reactive oxygen and carbon radicals and highly reactive species, and the latter is postulated to the lethal to parasite. When the reduction of this artemisinin to dihydroartemisinin, asymmetric carbon forms, it is possible to form oil soluble and water soluble prodrugs. When these drugs, oil soluble and water soluble drugs are there, the structural difference between this artemisinin, this artemether and artemoil and artesunate is that when this is methoxy group is substituted on this oxygen is replaced with the methoxy. This is also hydroxy with, and the remaining is the same one. This oxygen is, in case of this oxygen is replaced with this ester, it form artesunate, which is water soluble drug is developed. And when we substitute this R at this position, the CH3 and C2H5, it is artem till oil soluble and oil soluble. These two drugs artemether, this is OCH3 and OC2H5. This will be formed in water oil soluble agents from the artemisinin. The mode of action of again this artemisinin appears to kill the parasite by free radical mechanism, not by the generation of this selective or reactive oxygen species, but rather it by virtue of the free radical associated with the endoperoxide possibly involves the carbon radical. Just I discussed, it formed the radicals and the carbon radicals are there. It proposed that the heme in the form of amezoin within the digestive vacuoles is a source of this ferrous, which reacts with the peroxide to generate oxy radical and this ferrous. Again, this structural activity relationship, as we discussed, this artemisinin serve as a lead compound for the development of this antimalarial agent. This form reduction of this artemisinin to form this dihydroartemisinin. The hydroxy group can be alkylated. This hydroxy group can be alkylated. This hydroxy group can be alkylated to form oil soluble ether derivatives, isn't it? That is artemether and artemether. Then esterification of this hydroxy group is with succinic acid gives the water soluble derivative, that is artesunate. As we discussed, this is the artesunate, that is ester of this, and if it is what hydroxy group substituted with alkyl, isn't it? H is replaced with the alkyl, it form artemether and this artether, that is oil soluble, and this artesunate is the water soluble. The ester is water soluble drug, which is important for this biological activity. And the next class is this bigonides. These bigonides and the two drugs atovaquone and progonil. These two drugs are there. These are the structures of this. This progonil, in which the progonil reduces the effective concentration of this atovaquone needed to damage the mitochondrial membrane, and atovaquone increases the effectiveness of this progonil but not the active metabolites. The active form of progonil which acts as a dihydrofolate reductase inhibitor. When this progonil is a prodrug, isn't it? So when it, this is the mode of action or metabolism of this progonil, that is chlorovanide in presence of this cytochrome 450, it reduces, it is metabolized to cyclogonil by primarily by this cytochrome P2C19. The polymorphic nature of this hepatic enzyme explains subpopulations do not respond to this progonil, and these groups cannot convert the progonil to active form of the cyclogonil. On the basis of this, cyclogonil interferes with this deoxythymidate synthase by inhibiting dihydrofolate reductase, which is resistance to progonil. It attributed to amino acid changes, isn't it? So this atovaquone and progonil means progonil is a prodrug after metabolism it gets converted into cycloguanyl, that is active metabolite of proguanyl. This proguanyl is also called as the chloroguanide, isn't it? And atovaquone and proguanyl are administered in combination in the ratio of 2.5 atovaquone to 1 proguanyl, measured in the milligram, isn't it? And these drugs are developed in 1945, and it is an example of the prodrug. I repeat again, this is the prodrug proguanyl when it converts into active form in presence of enzyme CYP2C19, and this enzyme is responsible for conversion of this proguanyl to active metabolite called as the cycloguanyl. These are the miscellaneous agents of antimalarial: haloprantine, mefloquine, it is methanol quinoline, lumefantrine. This 2-quinoline methanol, this is mefloquine, is effective in the treatment and prophylaxis of this malaria, and it has a strong blood seasonticidal activity against Plasmodium falciparum and Vivex, and active against and not active against the hepatic stages or gametocytes. The adverse effect of this mefloquine is bitter in taste, and the higher doses it causes the nausea, vomiting, diarrhea, and abdominal pain. And the some drugs and antimalarial drugs, that is antibiotic antimalarials, are we will discuss in the antibiotic chapter. That is doxycycline and polycyclic antimalarials are there. Example is doxycycline, etc. drugs. This is regarding the remaining part of this antimalarial agent. Thank you very much.